I'll put the link back on. Oh,
Oh my gosh.
I haven't heard a lot of talk yet, but you look at letter A. If they have a kid with O blood type, what does that have to mean about their genotypes? Yeah, they have to be heterozygous, right? They both have to have that recessive I in order to have a child with O blood type. So their genotypes have to be I, AI, and I, BI, right? So then you would just need to think about that cross. Okay, so write out whatever that phenotypic ratio would be. If I was grading this question, that's what I would look at first, if you have the right phenotype ratio. Square. So if you fill that in, you can see there's how many different genotypes, 4, and how many different phenotypes. In this case, 4, okay. This one, if you have the A allele and the B allele, that child's going to have AB blood type. If you have IB little I, you're going to have B. If you have IA little I, you're going to have A blood type. And if you have 2 little i's, you're going to have O, right? So you could do 1/4, 1/4, 1/4. That's 1 to 1 to 1 to 1, right? So that's the kind of thing you need to be able to do with those codominant problems, and hopefully we're able to use those alleles to not get confused with the subscripts. Just treat them like you would a capital versus a lowercase letter, right? Questions on any of that? Sometimes when everybody's so quiet, I don't know if it's because it's like just so super easy. You feel like I'm like. Anyway. Okay, so on that quick check worksheet, any problems you want to take a look at? Or just pick a few. Number 4. Okay, so I probably will not go through all of number 4, but we could definitely do a couple squares. Do that right, so I'll put that in that one. So this is a multi-allelic trait, so we have multiple alleles that are going to affect the coat color here and the gene, right? Which one's dominant over all of them? The big C, which is a Goody. Which one's the next most dominant gene? CCH, which is chinchilla. And then which one's the next? The Himalayan. And then the 4th allele is recessive to all of them, right? And that is little c, which is the albino allele. Okay, so we have that kind of order of dominance, right? If you have a capital C in anything else, it's going to be a Goody. If you have CCH in any of these two, it's going to be chinchilla. If you have CCH in C, it's going to be Himalayan, right? If you only have two little C's, then it's going to be albino. Okay, so that's, if you read that carefully, that's important to know before you can figure out any of the phenotypes. So let's do letter B. So if we do letter B, we're starting with this parent who would have what coat color, what phenotype? Would be a Goody, right? Because they have the dominant C. And they are heterozygous for that in the chinchilla allele. The other parent is also a Goody, but they're heterozygous for a Goody and the albino allele. So if you fill in that square, you would get something like this. If it's helpful, you can put the slashes between them. A lot of times we put the slashes just to denote that difference where the line, where one ends and the other one begins. So 3 of them have the most dominant allele, the Goody allele. So out of those offspring, 3 out of 4 are going to be a Goody. And the other 1/4 is going to be chinchilla. Yeah, I probably should have flipped when I wrote the way that I wrote that. And then let's go ahead and do D also. We'll do D and D. So D, parent 1 is a Goody. They're heterozygous, just like this one, for the Goody allele and the chinchilla. And parent 2 is Himalayan, right? They're heterozygous for the Himalayan allele and the albino allele. And you don't have to put that slash mark there, but it just kind of helps you to like differentiate. This is the end right there. So if you look at that case, these both have the capital C, the most dominant allele. So 2 out of 4, 1/2 are going to be a Goody. And in this case, the other half both have the next most dominant allele, which is chinchilla. So those are both going to be chinchilla. And so in all this process, you just have to be really careful when you look at the offspring phenotypes, like where do they fall in that category of dominance, right? So what's their final phenotype going to be? So I think that's the trick to that one. Feel pretty good on that? I kind of like to go over number 3, I think, with you. So number 3, we're dealing with an epistatic gene. So we're adding on to the idea of our blood type alleles, right? So we're dealing with our ABO blood type alleles, but there's also this B gene, and that codes for the precursor to any antigen. So as long as you have one working copy, one dominant B, you can make antigens, and therefore you'll make whatever antigens the other gene codes for. Right? If you're homozygous recessive, you can't make any antigens, period, So it doesn't matter if you have the IA or the IB or the little I allele, you're always going to have what blood type? Oh, okay, that's why it's epistatic, because that gene, if it's homozygous recessive, is going to mask the other antigen allele. Okay, so for A, this individual is going to have what blood type? AB. They can make antigens, and they have the A and B antigen alleles. Okay. This individual is going to be type B, because they can make antigens, and they have the B antigen allele. This individual is going to be type O. Even though they have the A antigen alleles, they can't make them, okay, because they only have the recessive copy of the epistatic gene. Okay, so we get, does that make that, like, that's pretty important before we move on. Okay, so letter B then, we have to find some phenotypic probabilities for a cross between two parents. It really makes the problem a lot easier if we really focus strictly on phenotype ratios. So if I'm thinking about a Fort's line diagram, right, and I'm thinking about each trait individually, if I have individuals that are heterozygous for the antigen-making allele in the first place, what's my phenotypic ratio going to end up being? And we can make a square if we need to, right? B. Well, the genotypic ratio is 1 to 2 to 1, but the phenotypic ratio is 3 to 1, right? I'm going to have 3/4 that have at least one working copy, and 1/4 that are going to have both broken copies. And I can do a similar thing then with my blood type alleles. If I have a parent that is heterozygous B type, and I have a parent that is, I wrote it that way in the problem, heterozygous AB, I can look at that cross. IA, IB, IA little I, IA big B, IB, IA little I. Oh, well, that's all right. Sorry. Okay, so I have 4 different genotypes, but actually how many phenotypes? 3. 1 out of 4 is going to be IA, IB, or AB, if you would want to put AB. 1/4 is going to be, well, it's going to be A. And I have 2/4 that are going to be B type. So then if we're making a Fort's line diagram, right, I can multiply all the possibilities for this cross, all the possibilities for this cross. I'm just going to simplify it here. 1/4 AB, 1/4 A, 2/4. And this technically you don't have to do because you know right away all the rest of the offspring are going to be type. But we'll do it anyway. 1/4. Of course. Okay. 3/4 times 1/4 is 3/16. And if you have a working copy and you have the AB alleles, those offspring are all going to have what blood type? AB. All right. 3/4 times 1/4 is 3/16 are going to have type A blood. 6/16 are going to have B. All the rest of these individuals are going to have type O. So you could just add up 3 plus 3 plus 6, which is 12. Subtract it from 16. That's how many are going to be type O. But just so 1/16, 1/16, 2/16. All of those individuals are going to be type O. So there's my phenotypic ratio, 3:3 to 6:4. Questions on that? How many of you got that ratio correct? 2 people? Okay. So I think that's kind of I think that is a harder problem. Like there's a lot going on there. So didn't get it right the first time. Okay, but any questions on that before? Any questions on the front? I think one was really easy, so I don't think we probably need to go over one. There are some mistakes I see commonly in number 2. So you really did need to set up a Punnett square, I feel like, for the cross, but mom's AB, dad is A, but his dad was O. So why is that important? Yeah, it tells you that he's going to be heterozygous for that A blood type, right? So he has one A allele and one O allele, right? One of the recessive alleles. Okay, so that's pretty important to know. Okay, that's why that information is included there. So, use Punnett square. I. The phenotypic ratio is going to be 2/4 A to 1/4 AB to 1/4 B, right? And so you need that information then to calculate all the probabilities. So A, so what's the probability that 2 children will both be a group A? Every child is a unique chance, okay? So you have to multiply each possible probability. So what's the chance that child A will be group A? Did I set that up right? Yeah. It's going to be 2/4, right? Or 1/2. We can just say 1/2. And what's the chance that the other child will be group A? 1/2. So the probability that both children are going to be group A is 1/4. Or if you didn't simplify it, it would have been 4/8, which reduces, or 4/16, which reduces down to 1/4. Questions on that? One child will be group B, that's 1/4. One child will be type O, 0. Okay, 0 chance. Letter C. Not only are they asking about blood type here, but now they're talking about gender. That's a whole extra probability, right? So, first of all, the first child will be a son. What's the probability of son versus daughter? 1/2. What's the chance that that son's going to have AB? 1/4. What's the chance that their next child's going to be a son? What's the chance that that son's going to be group B? 1/4. So 2 times 4 is 8, times 2 is 16, times 4 is 164. questions on that. So you can do all kinds of probabilities just by multiplying each singular probability together, right? That's kind of the point of that problem. All right, so if you didn't get that turned on Canvas, please make sure you get and if you fixed anything, you can resubmit. Okay, try to get that in tonight. We do have one last really important non-Mendelian pattern to look at today, and that's sex-linked traits. So, sex-linked traits are traits that are located specifically on sex chromosomes. And just a reminder, what are the sex chromosomes for humans? X and Y. As opposed to genes that are on autosomal chromosomes, or any of the other chromosomes, right? Any other 44 chromosomes are all autosomes. Only the X or X and Y chromosomes that you have are the sex chromosomes. Okay, so these genes are either going to be on the X chromosome, mostly the X chromosome. Talk about why that is. These were first discovered by T.H. Morgan. He did a ton of important work with fruit flies. Really helped our understanding of genetics. If you ever take a genetics class in college, you'll have to do general fruit fly protein. The scientific name for those fruit flies are Drosophila. So, why do you think we would use flies as our organism to model genetics? I'm going to talk to your groups really quickly. Why do you think they'd be good organisms for that? Yeah, they're relatively cheap. What else? What else would they be good to use? They're fairly easy. I mean, they're small. But yeah, we can look at them under a dissection scope pretty easily. They reproduce really quickly. Have lots of offspring. So they're prolific. Yeah, they have lots of offspring all the time. They're pretty short turnaround generation-wise, but they only have 4 pairs of chromosomes, so they don't have that many chromosomes to work with. And also, they do have the similar sex chromosomes to us, whereas this is actually more complicated than that. It's not quite like ours. But they also have XX chromosomes, females, and XY chromosomes. And they're just a little bit more complex than we would like that. So, this is just a really quick review on the difference between autosomal chromosomes and sex chromosomes. So again, sex chromosomes are only X and Y, whereas all the other chromosomes are considered to be autosomal. Okay? So a lot of times when we talk about human genetic diseases, we will indicate whether they're autosomal or whether they're sex-linked. We get to categories a little bit later next week. You'll have to differentiate between whether a trait's autosomal or sex-linked. Okay? So autosomal, everything else, sex chromosomes, X and Y. And so here's the kind of famous T.H. Morgan experiment that kind of led to us discovering sex chromosome linkage. So just like Mendel, he followed the same pattern where we have a parental generation and then F1 and then F2. So we're crossing true breeding parents in the P generation, a red-eyed female, and a white-eyed male. red is the wild type dominant trait, white is the mutant recessive trait. Okay. So when we do those crosses, what would you expect the F1 generation to look like? All red eyes, yep, and that is what happened. 100% red eye offspring. All the males and all the females have red eyes. What ratio would you expect then when we cross those F1 generation? You have a dominant recessive trait and you cross two heterozygous parents. What's the phenotype ratio? 1 to 2 to 1. Well, genotype's 1 to 2 to 1. Phenotype is 3 to 1, right? So you would expect 3 to 1 ratio. And we did get a 3 to 1 ratio, but what was unusual is all of the females had red eyes. Okay? There's no 3 to 1 ratio in the females. They all had red eyes. Okay? The males were 1 to 1. Okay. So overall, we had a 3 to 1 ratio, but this was really, this is pretty striking that all of the females have a dominant trait and the males are 1 to 1. So unusual, not following what we would expect to see with Mendelian genetics. So we know something's going on here. And so we're going to look at a couple Punnett squares really quickly. Again, this is what we would expect if it was following normal patterns, right? So here's again the parental generation. We would expect all of them to be heterozygous, right? And then if we cross those F1 generations, we would expect that 3 to 1 ratio, okay? But this doesn't explain why all the females were red and all the males were either one or the other. So there has to be something going on here besides being on a regular autosomal chromosome. Okay? So that's not what happened. So this is something we've already mentioned, so we'll go through this pretty quickly. But in humans and other mammals, there are 2 sex chromosomes, X and Y. You have 2 X chromosomes, you are female, and you have an X and a Y, and so you develop as female. If you have, what this also means is that just like all of our other autosomes, we have gene redundancy, which means we have 2 copies, right? If one copy doesn't work and the other copy might, okay, you're still going to show the dominant trait. You're going to show the working copy of the protein. If you have an X and a Y chromosome, then you develop as male, but you do not have that redundancy. So if you have the broken copy, you're SOL, right? You don't have a backup copy to provide the working version of the gene. Okay. So when we set up any Punnett square dealing with these chromosomes, we're going to have XY on one side because that's going to represent dad, and you're going to have an XX on the other side to represent mom. And when we deal with these specific traits, we'll have some superscripts then on those X. If it's on the X chromosome, on the X chromosome, okay, that will denote what version of the X chromosome we're getting. But anytime you do the cross between mom and dad, there's always going to be a 50-50 chance of being female and a 50-50 chance of being male. So 50% chance female, 50% chance male. So let's go back and take a look at those slides again. This time, we're going to think about the genes being on the X chromosome. So notice, mom's a true breeding red-eyed fly, which means she has both dominant alleles on her X chromosome. So X superscript R, X superscript capital R. Okay, 2 dominant versions. Dad is XY. He only has one copy of that R allele, and what's that letter going to be? Little r, okay, because it's recessive. And he has a white eye. So do me a favor and set up that Punnett square on your paper there. What should that look like? You do that. So if we were going to do a genotype ratio over this, the genotype ratio would be 2/4 to 2/4 or 1/2 to 1/2, right? There's only two genotypes. When we think about phenotype, we want to specify male and female, right? So we have half females with red eyes and half male with red eyes. Okay? Even though they all have red eyes, we want to make that distinction on these sex-linked traits. When we take these parents and cross them in the F2 generation, go ahead and set up that Punnett square. Now we have how many different genotypes? Well, technically have 4 different genotypes, right? We have female homozygous dominant, we have female heterozygous, we have male dominant, we have male recessive. Okay, so 4 different genotypes. Phenotypically, 2/4 are female with red eyes, 1/4 male with red eyes, 1/4 male with white eyes. So if you're a male, you're only getting one X chromosome. So your mother is very important in determining all of your X-linked traits, right? Because you don't get an X from your dad, you only get your X from your mom. Okay. If you're a girl, you're always going to get dad's X-linked trait and then one of mom. Okay, so that really leads to some interesting patterns in families that when we get to our pedigrees in a little bit, we will see, again, some interesting patterns based on sex-linked traits. So, really important when we're dealing with these that you're keeping track of X and Y chromosomes and whether the genes on them are the dominant or recessive copies. So, Y chromosome is a pretty small chromosome. There are actually very few genes on it, other than the SRY gene, which is the vital gene to determining sex. Okay, so if you have the Y chromosome, you have this SRY gene. And that tells your reproductive system to develop as male. If you have the gene, you're going to be male. If you don't have the gene, the way that you develop is female. So female is kind of the default pathway. And then if you have SRY, then it changes it to male instead. And why do you think there's so few genes on this Y chromosome? Do females have a Y chromosome? No. So if there are really important genes on there, we wouldn't be able to be here, right? So almost all of the important genes are on the X chromosome. So there's lots of other genes and traits beyond sex determination that are on the X chromosome. So some of these mutations can cause issues with hemophilia. Do you guys know what hemophilia is? What disease is it? It's a blood disorder. Yeah. It's a blood disorder where you have trouble clotting. So if you get cut, you don't stop bleeding properly. Okay, so that is obviously a big issue. It also means you can have strokes much more easily and stuff like that. There's the clotting factor, one of your clotting factors. Duchenne muscular dystrophy, so that's where your muscles kind of waste away and stuff properly. Color blindness, in our day and age, not usually so lethal unless maybe you have issues with stoplights. But so a lot of color blindness has to do with genes that are located on the X chromosome. So X chromosome is a very large chromosome, has lots of really important genes on it, and that's why both males and females have that one working copy of the X chromosome. So those are some pretty important things. So generally, when we're talking about sex-linked traits, it usually means, not always, that the trait is X-linked. So more than 60 diseases are traced to genes on the X chromosome. And I know that's, again, really tiny, but again, we talked about muscular dystrophy up here. Fragile X might be one you've heard of. There's a lot of eye stuff on here. Some metabolism-linked traits. All kinds of different things. Hemophilia is down here towards the bottom. Diabetes insipidus is on here. So all kinds of different genes that are important. Color blindness is down here towards the bottom. We take a look at the human Y chromosome, only about less than 30 genes. And again, none of these tend to be, so this is obviously funny. But there is one gene, again, that's really important, which is that SRY sex determining region. Okay. Hopefully nobody's super offended by that. I didn't make this slide, but I just put it in. So we tend to see this trait. So hemophilia is an example. So here's a pedigree. We'll get to these. Hopefully, do you kind of remember pedigrees at all from Bio 1? A little bit. So you'll notice, for example, that anytime you have a mom that's a carrier, she's going to be more likely to have sons that have hemophilia, right? Because they're only getting one X chromosome. So if mom has that defective copy, there's a one out of there's a 1/2 chance they're going to get that defective copy, right? And therefore have hemophilia. They don't have that backup copy. And so we'll take more of a look at that later. So, but just like with our fruit flies, it's important when we write out these chi-squares that we use the X and the Y, and then we use our superscripts to denote whether the allele on the X chromosome is dominant or recessive. We do have a special name for the female who's heterozygous. We call her a carrier because she's carrying that recessive trait, but she's not going to show it, right, if it's a recessive disease. Okay, so if you haven't heard the term carrier before, that's kind of an important term. Okay, so I want you to try these practice problems real quick. Color blindness is due to the X-linked recessive allele B, while the X-linked dominant allele leads to full color vision. Predict the ratio of offspring phenotypes for each of the following. So for A, you have a colorblind female. What would her genotype have to be in order to be colorblind? X little, whatever letter you want to use. What do we say? C, XD, XD. And male with full color vision, so he would be X. He'd have the dominant copy if he was not colorblind. So part of the, I think the hardest part of this is maybe making sure you're starting with the right genotype. So go ahead, try and discuss those problems with your group really quickly. give you like 5 minutes to leave. So you probably can't read that. But if you do this first cross, all the girls are going to be heterozygous, so they're going to have normal vision, right? All of the males are going to have only the recessive allele, which means they're going to be colorblind, right? So if your mom's affected, the son's going to have it no matter what, right? The only chance he has is getting the defective copy from mom. Yeah, so I think a really important thing to take away from sex-linked traits is that in order to have a recessive sex-linked trait, if you're a male, you have to get it from mom. Okay? If you're a female, you have to get the defective copy from both mom and dad. Okay, so that causes a lot of the differences between the phenotype ratios you'll see in these kind of traits. So. And that class period's almost over. We did not quite get there as much as I wanted to, but that's okay. So we'll have a little bit to wrap up.